Yeah.
Sam's going to be talking about tolerance to endogenous opioid tone causing persistent reinstatement of hypersensitivity in a mouse model of inflammation. Over to you, Sam.
Thank you. I'm assuming my slides are shown. I can't see anything apart from my screen. But my name is Sam, and I'm a final year PhD student here in Dundee, working under the supervision of Professor Tim Hales. And this is some of our preclinical work looking at the role of tolerance to endogenous tone in the development of persistent pain. So in our bodies, there are three main types of opioid receptors. These are the mu, delta, and the kappa receptors. We also have endogenous opioids, which act at these receptors to modulate pain by promoting antinociception. These are also the same receptors that clinically use analgesics, such as morphine, bind to. And you'll see here that the primary receptor for these analgesics are the mu receptor. And activation of these is associated with a number of side effects, including respiratory depression, constipation, and tolerance, which requires escalating doses to achieve equivalent pain management. And these receptors are called G protein coupled receptors, and they couple to inhibitory G proteins, meaning that once the receptor has been activated, it leads to the activation of intracellular pathways, which reduce the cell's activity.
Sorry to interrupt you, Sam. It's a little difficult to pick up the sound. Is there any way you can speak into the microphone a bit closer to your face?
I can come in close. I'm sorry for the view, though. In neurons, this is achieved by inhibition of calcium channels and also by activating potassium channels. And this causes less current to come in and more to go out, leading to reduced activity. Over time, these receptors will also be phosphorylated, which leads to the recruitment of these proteins called beta-arrestins. And beta-arrestins are extremely important in the development of tolerance. And so we can measure this using relatively simplistic tools, such as the tail withdrawal assay, which measures the latency of a mouse to withdraw its tail before and after morphine, which we can monitor over several days. And if you repeatedly give a mouse morphine, then it will quickly develop tolerance. So the tail withdrawal latency will get shorter and shorter, although this doesn't happen if you knock out beta-arrestin 2. And knocking out beta-arrestin 2 is also being reported to improve some of the other side effects, such as constipation and respiratory depression, although this is currently debated. And while tail withdrawal assays are very good for measuring simple responses, they're not a particularly good pain model, and so it's hard to generalize those findings. And in this example, mice have been given a compound called complete Freund's adjuvant, the CFA, which causes chronic inflammation. And as you can see, their withdrawal thresholds, so this mechanical sensitivity, were measured before and at several time points after CFA, and they decrease following the injection, and so they become hypersensitive. And this hypersensitivity recovers after about 2 weeks. However, when mice are injected with the compound called naltrexone, NTX, which stops any activity at the mu receptor, then this hypersensitivity is reinstated for many months, telling us that mu receptor activity is involved in recovery and that this may last indefinitely. However, over time, the ability to recover is reduced, which may be indicative of tolerance to endogenous mechanisms. And there are many different theories on how tolerance develop, and this by no means an exhaustive list. But this page just summarizes that because there are so many things that influence the development of tolerance, we have a lot of techniques that we can use to manipulate the development of tolerance, including genetic, biochemical, and pharmacological approaches. And with these approaches in mind, we attempted to alter the reinstatement of hypersensitivity by manipulating the development of tolerance and mu receptor function. And so first, looking at the role of mu receptors, then we can hypothesize that if mu receptor activity is required for recovery from hypersensitivity, then mice which have these receptors deleted should not recover. And we can see this comparison here. On the left is looking at the mechanical sensitivities of normal wild-type mice, while on the right is the same mice lacking mu receptors. And while hypersensitivity was transient in the wild-type mice on the left, it was persistent in mu knockouts. And these mice also just did not recover, suggesting that mu receptor function is required for recovery. We then use quantitative PCR to determine whether there are any changes in genes involved in endogenous analgesic tone within the spinal cords of mice that have recovered from the mechanical hypersensitivity. And we found that there was a significant increase in delta opioid receptor expression without any changes in mu receptor function expression. And there are also changes in the expression of kappa receptors and all of the endogenous peptides, suggesting that inflammation promotes changes to endogenous opioid tone. So following injury, there's an upregulation in endogenous analgesic mechanisms, which become enhanced to compensate for the development of pain hypersensitivity. And then we repeated these experiments in mice where the mu receptor was deleted, and we found that there was now no change in the expression of any genes involved in endogenous tone, suggesting that mu receptor activity is promoting changes to endogenous opioid tone. And without this mu activity, there's little ability to compensate for pain hypersensitivity. And we also repeated these experiments in delta knockout mice, which we showed on the previous screen were significantly enhanced during inflammation, and found that these mice also had limited abilities to alter endogenous opioid tone, suggesting that both mu and delta receptors are required. And having shown that both of these receptors are required, we looked in a cell model overexpressing mu receptors, which we then transfected with delta receptors to try and replicate the changes in receptor expression occurring during inflammation. And then we measured how this affects mu receptor signaling to beta-arrestin 2 and also to inhibitory G proteins. And we found that more beta-arrestin 2 is recruited to the mu receptor in the presence of delta receptors, and co-expressing mu and delta receptors in the same cells also causes inhibition of cyclic AMP accumulation, suggesting that there's some constitutive activity going on within these cells expressing both receptors. And this we know also alters the pattern of kinase activation, such as persistent ERK phosphorylation. And so we took this back into mice to see the effect of deleting the delta receptor, and found that this considerably delays the recovery from hypersensitivity shown underneath. So mice take about twice as long now to recover. And previous literature also suggests that the delta receptor is required for mu receptor function. And so if you take a neuron from a wild type and a delta knockout mouse and you treat them with an opioid, then these opioids have reduced effects in the knockout animals. So these studies suggest that the role of upregulating delta receptors within the spinal cord is to initially help these mice to recover. So we then wanted to promote the development of tolerance to see how this altered the hypersensitivity. And by giving mice daily morphine to promote the development of tolerance, we saw this initial recovery followed by a period of reinstatement of hypersensitivity, suggesting that tolerance to the endogenous tone caused by morphine enhances pain hypersensitivity. And then we verified that this was caused by tolerance by using mice which expressed only 50% of the mu receptors, which we've previously shown accelerates the rate of tolerance, and consistent with previous reports that we see down here on the left, the rate of enhancing the rate of tolerance also accelerated the reinstatement of hypersensitivity in this chronic model of inflammation. In contrast, if we gave a very high dose of morphine to beta-arrestin 2 knockout animals, which I mentioned did not develop the tolerance, then we delayed the reinstatement of hypersensitivity, and we could accelerate the rate of development of hypersensitivity in wild-type animals compared to the lower doses of morphine. And so putting this all together into a working hypothesis, we suggest that an injury leads to the good inflammatory response, although this will have pathological consequences, such as pain hypersensitivity. And this sensitivity or hypersensitivity is resolved through mu receptor function, which may then cause tolerance directly due to the increased activity of mu receptors. But mu function will also alter the expression of genes involved in endogenous opioid tone, and also the pattern of kinase activation, both of which will feed back onto the pathological pain, leading to priming of the mu receptors for tolerance. And once this is then established, persistent hypersensitivity is reinstated. Tolerance, you accelerate the reinstatement, whereas if you reduce tolerance, you can delay or avoid reinstatement altogether. And some of the other work ongoing in our lab is looking at some of the potential mechanisms through which we can do this, including targeting beta-arrestin 2 delta receptors or some certain kinases such as SPaRC. So I think for the interest of time, I'll stop there. And I'd just like to thank Tim and also Dan and Claire for aiding in the work, and also to the SPaRC committee and to Jackie for organising it and all of the helpful comments. Thank you.
Thank you very much, Sam. Fascinating. Are there any questions from the audience? Please type them in now, if so. We have very limited time. Sam, I wondered whether you thought there was any mileage in the use of either delta or kappa opioids using the same sort of interactions with arrestin and SPaRC, whether you could manipulate that either on their own or with mu opioids from the work that you're carrying out.
Yeah, that's an interesting hypothesis, but none of the clinically used opioids are delta or kappa typical agonists, and that's because these opioid receptors are associated with perhaps even more detrimental side effects. So delta agonists cause seizures, and kappa agonists are quite dysphoric. However, there is significant interest in the role of potentially what are called biased agonists, particularly for the delta opioid receptor in models of chronic pain. And these are ones which avoid the recruitment of beta-arrestin 2 and activate the G protein pathways. And there's significant interest in whether or not these can cause antinociception without any of the tolerance and any of the seizures that are typically associated with delta use.
Thank you very much.
However, the role of bias is controversial.
Is there anyone else wanting to ask a question? We are very short of time. So with that, because there's nothing cropping up on the chat, I'm going to leave it there, Sam, and many thanks. And hopefully move on to Kieran Dinwoody, who is hoping, I think, to be here by now. Kieran. Hello.
Hello.
Right, Kieran, it's wonderful to have you, and you're going to give an update on the national